Okay, this will be the discussion for Lab 24. That's the ECG and Pulse Laboratory. As a quick reminder, the heart beats as an integrated unit. We call that a functional syncytium. A syncytium is a network of interconnected cells. It's the intercalated discs that allow that to happen. Most of your heart is cardiac muscle, and it's those interconnected cells, the intercalated discs, that allow electrical signals to pass directly from one adjacent cell to another. Although, as discussed in lecture, the atria don't pass the electrical signals directly to the ventricle. That's part of the conduction system. We won't worry about the conduction system for the most part here, though. The electrical signals come from autorhythmic cells, right? Sometimes known as the pacemaker cells. Those are cells that can generate their own action potentials. They're part of the cardiac nodes. The cardiac nodes that you find in the heart are the SA node, the sinoatrial node, the AV node, which is the atrioventricular node, and then the V nodes, which are the ventricular nodes. So those all can possibly give rise to their own action potentials. However, under normal circumstances, it's the SA node that sets the rhythm for the heart. So that's called the pacemaker, typically. Just as a quick reminder, the SA node is in the upper right portion of the right atrium. The AV node is at the bottom of the right atrium, right on the junction, and the ventricular nodes are scattered throughout the ventricles. The electrical conduction, just in a nutshell, we're not going to worry about it in lab. We'll kind of cover it in lecture a lot more, but as a reminder, the normal circumstance is the SA node generates an action potential that travels through the atria, that gets picked up by the AV node. The AV node then transfers it to the bundle of His. The bundle of His branches off into the left and right bundle branches. Those go down to the apex of the heart, that pointy part at the bottom. They turn up. They turn into what are called Purkinje fibers, and those are what releases the action potential to the ventricular muscle. So the atria basically contract from the top down and push blood into the ventricles. The ventricles contract from the bottom up and push blood into the large arteries exiting the heart, the aorta or the pulmonary trunk. So we can measure that electrical activity with what is known as an electrocardiogram, sometimes called an ECG, or alternatively an electrocardiogram with a K called an EKG. We see in the United States more often than EKG, but you still see EKG, and it's supposedly a remnant when medical orders were given orally, and there's difficult to say, confuse ECG with another medical test, an EEG, right? So EKG, yeah, it's pretty different, but ECG, EEG are similar sounding, and even written looks similar. So that's one of the reasons why it's used in the United States, even though we typically spell cardio with a C. So it's a recording of the electrical activity of your heart. It uses a principle called volume conductance, and that means the electrolytes pass the electricity through the different tissues. We can measure the ECG very simply. We measure what is sometimes known as a modified lead 2, or sometimes called a V5. We have an electrode, a negative electrode, that's the white one on the right wrist. We have a positive electrode on the left leg, and then we have ground on the right leg, and we measure the electrical activity across the heart that way, and that gives us the typical ECG pattern we'll look at. So volume conductance allows us to measure the different parts of the heart at the limbs. It's a very diagnostic procedure. We use it primarily to measure heart rate, but you can see if someone had previous heart attacks. You can see if someone has conduction abnormalities, because that conduction system I just briefly explained. You can see if the various chambers of the heart are different sizes. You can see what direction the heart is pointing. Very useful and diagnostic tool, and it's great because it's non-invasive. It's extremely safe. So this is the basic heart rhythm. It's a series of waves. We have a P wave, then a Q, then an R, then an S, then a T. so PQRST. Each one of those waves corresponds to various portions of the cardiac cycle. So the P wave, right, so this little first one, that corresponds to atrial depolarization. So you have a P wave if the SA node is firing. And so we see that electrical activity from the SA node in the P wave. If you did not have a functional SA node, you would not have a P wave. Okay? So that's the P, that's atrial depolarization. That's the electricity that causes the atria to contract and push that last 1/3 of blood into the ventricles. We also have the QRS. Now the QRS we often put together as a complex, so it's called the QRS complex, and that corresponds to two important events electrically. The first is, since the P wave, right, that's electrical activity of the atria, eventually gets passed through the bundle of His and the left and right bundle branches, and so the resulting wave complex that we see, QRS complex, corresponds to ventricular depolarization. This is when the electricity leads to ventricular contraction. In addition, hidden in this larger wave is the atria repolarizing. Remember that once tissue depolarizes, it has to repolarize. It has to reset so it can fire again. So two things happen in the QRS complex. And then in the T wave, which is this larger wave, that's ventricular repolarization. So that's the ventricles repolarizing. Remember again that the actual electricity precedes the contraction and actually causes the contraction. So if you remember kind of the order of the different waves and complexes, P, QRS, and T, and you remember that we start with the SA node, then it's pretty easy to remember the order which happens. The other thing I do is I just remember, okay, one thing happens for the P wave, atrial depolarization. Since the QRS is a complex, it's multiple waves, multiple things happen, ventricular depolarization as well as atrial repolarization. And then for the T wave, we get ventricular repolarization. So that's the different events that happen during various waves of the cardiac cycle. The other thing to kind of remember, well, remember because I don't think I've told you yet, the actual action potentials for the atria and the ventricle look a little differently. So we're not measuring the action potentials, we're just measuring electrical activity. So the action potentials look like this bottom right, which we're not going to worry about at all in this lab. So looking at the electrocardiogram or the ECG, you can see it's just a series of waves that just keep on going over and over again. By definition, your whole life, right? Otherwise, if your heart's not beating, right, you're not living. So the way we measure heart rate is we use what's called the R to R interval. So again, we've just shown the major waves that are going in a positive deflection in the way we're looking at the heart. And so we measure what's called the R to R interval. So you measure the peak of R and you measure the time it takes to get to the peak of the next R. And when you measure that, then you can figure out how long, how many milliseconds it took between beats, and then you divide 60 by that number, and that'll give you your heart rate. We said the normal heart rate is about 60 to 100 beats per minute. If it's slower than 60, that's called bradycardia, right? The causes would be a genetic abnormality, or just not even abnormality, just a genetic difference. You might have a slow SA node. You could have a very active parasympathetic system that's doing that. You could have a very good body conditioning state where exercise has caused your heart to become stronger, so you have an increased stroke volume, or it could be caused by disease. So some disease processes also lead to a slowing in the heart rate. Tachycardia, right, would be a heart rate above 100. Again, it could be a fast genetic SA node. It could be related to a very inactive parasympathetic nervous system at rest. Could be other things as well. Body size, your gender, disease states, all can lead to tachycardia as well. ECGs are very diagnostic. Here's just some examples. Don't worry about it. I'm not going to ask you on an exam about these things, but this is just showing some differences. So here we have, P waves that don't always generate a QRS complex, so that's a problem with the conduction. Okay. Here we have random P waves that just occur all over the place. That's an even bigger problem with conduction and different phases of heart block. So those are the different things you can see. There's many different things you can see. And again, if you're going to need to understand ECGs in your job, if you're doing some sort of clinical thing, you'll have to certainly take an ECG class or multiple ones. Nowadays, many nurses are not required to understand ECGs as much as they used to in the past, because many of the machines, literally the machines that take ECGs, do the diagnosis. And then when it finds, potentially faulty rhythms, oftentimes it literally directly texts the doctor on call, the cardiologist, who looks at it and then makes a decision about what to do. So here's other examples of atrial or ventricular fibrillation where the muscle's not pumping. VFib is when you actually need to shock somebody and get them back into a normal rhythm. So with that background, let's sort of look at some of the things we looked at in lab. So first thing we'll look at is BioPack Lesson 5. So in BioPack Lesson 5, you would be responsible for the background information from the BioPack itself in Lesson 5. So when you look at the supplement in Lab 24, there's background information. You'd be responsible for that. And so what we would do here is we would just measure the R-to-R intervals I explained earlier, and just highlight this on the computer. If this was the BioPack tracing, it'd look a little different than this, but you can get the idea. And it would display the heart rate. So for our subject, right, the heart rate was 74, which is right on the average normal heart rate. You should know the normal range, 60 to 100, and you should know the terminology. If it was below 60, it would be tachycardia. If it was above 100, it would be bradycardia. So this is sort of our control heart rate that we're going to use to compare to other things. We also, in class, would measure the different sizes of the P wave and PR interval in all these different waves, intervals, and segments. We're not going to worry about all of them. We're going to worry about a few of them, but we're not going to worry about all of them. So don't memorize. So here's one of the other things that you should understand about this. We would have done the deep inhalation, deep exhalation while we're just sitting. So somebody would just take a deep breath in and then deep breath out. And one of the things that kind of want you to look at, and I like this figure a lot, is when the person takes a deep breath in, this is the inhalation, so you can kind of see the respiratory in that kind of lightly dotted line behind, and notice the ECG bunches together, all right? When they exhale, notice the ECG gets farther apart in terms of the R-to-R waves. So if you were to measure the heart rate for inhalation for this subject, it went up to 92. And then during exhalation, it went down to 60, 61. So what happens is during inhalation, a couple things happen. What we see is during inspiration, there's a active process that happens. You have an increased muscle movement, you have increased sympathetic activity, you have decreased parasympathetic activity. That's because you have an increase for more oxygen to go to the muscle because of that muscle movement, because inspiration is an active process. So what we do is we actually flip a little switch in our brain that causes our heart rate to go up. So heart rate goes up during inhalation, so we expect to see that go up to 90 beats per minute. That's pretty normal. During exhalation tends to be more passive. There's reduced muscle movement. You have a decreased, so this should say decreased sympathetic activity and an increased parasympathetic activity. And so sometimes this is called respiratory sinus arrhythmia. But for the data, it looks exactly the way we should. So inspiration heart rate should go up, expiration heart rate should go down because of those changes. So if we look at exercise then, we saw that our subject exercised, and then after exercise, we didn't want to exercise that hard, and the heart rate went to 125 once they finished. Okay, so we looked at that exercise heart rate. So one of the things we wanted to look at, though, during exercise is what happens to the QT and TQ interval. So going back, remember QT interval, which is measured as an estimate of ventricular systole, so when the heart is contracting, and it's best measured as the peak of R. Again, it's the peak of R because we can't measure when the electricity starts because it's the electricity that causes the contraction, right? And we want to measure the contraction itself. So we measure from the peak of R to the end of T. So it's called the QT interval, and it corresponds to ventricular systole. From the end of T to the next peak of the next R wave, that's sometimes called the TQ interval, and that measures the ventricular diastole. So notice ventricular systole, very short, and diastole about twice as long. And that's again where that 1/3 systolic plus 2/3 diastolic formula comes for mean arterial pressure. So at rest, the QT interval, right, is about 1/3 of, the easiest way to say it is the systole is about 1/3 of what the diastole is, comparatively. So as an example, we'll look at some data and see that you have this relatively short systolic time and this relatively long diastolic time. When you exercise, that actually changes, and your systolic time and your diastolic time come closer together. Okay? So exercise reduces diastolic time. You can't reduce systolic time too much. You can do a little bit, but it still physically takes time for the heart to contract. But for filling, we can decrease the filling time by increasing the pressure difference. And so at rest, right, you'll see this, fairly long systolic and diastolic time. So, systolic time. So here's my systolic time. If we just measure from here to here, right, really slow heart rate. And then all this right here is my diastolic time. All right, I'll highlight it for you, right? So if we go from about right here to right there, that's my systolic time. Do it in blue. And then if we go from here to right there, that's my diastolic time, right? So notice how short the systolic versus diastolic time is. If we come over here, since we're already on this color, here's my diastolic time in blue there, and then going back to that pink color, here's my systolic time there. So notice, yeah, the systolic time shortens a little bit, but proportionally, the diastolic time portion significantly decreases. So that's kind of looking at the difference between systolic and diastolic time. So during exercise, it goes to about half and half, and the harder you exercise, the greater degree the diastolic time decreases. If you're at maximum exercise, it actually flip-flops in most people and goes to 2/3 systolic and only 1/3 diastolic. So we can see that from some of the data we would have collected, as we can easily measure that on the BioPack machine. So here is our first subject where their time between beats, right, during sitting rest, so that's the R-to-R interval, was 0.81. Okay, so our subject had a time between beats of 0.81 seconds. So if you go 60 divided by 0.81, that's the heart rate of 74 that we saw at rest before. Okay, and then what we did is we measured the QT interval, which was 0.27, and we measured the QT interval, so that was 0.54. So the systolic time was 0.27. So 0.27 divided by 0.81 is exactly 1/3. We don't usually see this exact numbers, but this is kind of neat. This was one of the subjects we had a couple semesters ago. And then for the diastolic time was 0.54. That's exactly 2/3 of the 0.81. So this person at a heart rate of 74 had 1/3 systolic and 2/3 diastolic. Now when you look at their exercise, this was the heart rate of 125 we just saw a minute ago. All right, so their RDR interval time was 0.48. And then the systolic and diastolic time were exactly 0.24 and 0.24. So we went from 1/3, 2/3 to half and half. And had we continued to exercise this person even more and more strenuously, then they probably would have gone to something like at a heart rate of 180. It would have been, probably, let's see, let me kind of look at the math. So a heart rate of 180, that means the time for a beat, right, would be about 0.33 seconds. And so that means the systolic time would be probably like 0.22-ish, and the diastolic time would be 0.11. So we'd go, you can't decrease systolic time too much because physically it takes time for the heart to contract. But diastolic time we can decrease a significant amount of time. So that's something to remember here as well. So that finishes up the ECG only part. As a quick summary, the ECG, you should understand about the ECG in general. You should understand the waves and what they correspond to, the PQRST waves we talked about. You should understand what happens with inhalation and exhalation. You should understand what happens with the QT and TQ interval, the systolic and diastolic time at rest and during exercise. You should also understand the ECG and pulse portion of this. Right, so we kept the ECG on. I actually did this first in lab because this one is, the last one involved exercise. And in order to use the same subject and you want resting pulse, then it's best to do the exercise last. So that's why we did it that way. For the ECG and pulse, we're going to look at something called the pulse pressure wave. And we use what's called a plethysmograph. So you can see it over here. Plethysmograph's hard to say. It's another good hangman word if you play hangman. And what it does is it measures volume changes. So we're going to measure the volume change, and the blood volume change is going to give us the pulse pressure wave. And so when we took pulses the first week of school, the radial pulse, and measured heart rate, you can do it at the carotid pulse as well, then we're measuring that pressure wave. And the idea is the pressure wave should be a good indication of the heartbeat, and we can use that to measure pulse. And we know that is true because we clinically often measure pulse rates at the radial pulse, right? So what we're going to do is we're going to put that pulse pressure plethysmograph on your fingertip and measure the pad of your finger in terms of its volume changes from that pressure wave. And we're going to keep the ECG on the same way we had it on before and measure heart rate and look at the differences between those. So here's an example of someone where we have the arm rested on the table. So this is just normal rest. It's about heart height. And we're looking at the RDR interval. Remember, the RDR interval is the heartbeat. So it's 0.78. And at 0.78, if you do the calculations, it gives us a heart rate of about 77 beats per minute. We also measured the pulse interval. In measuring the pulse interval, we measured the peak of the pulse to the next peak of the pulse wave. And that was 0.77. And that gives us a pulse rate of 77.9. So in this case, the heart rate that we measure and the pulse rate that we measure are not exactly the same, but they're pretty close. And usually that's just measurement error. It could have something to do with the travel time of the pressure wave, though, because it is a physical thing and it's affected by gravity. So what we did is we do two different experiments. One, the person has their hand below the heart. You can put one of your hands in ice water. It doesn't tend to do much, but you can do that. We would just do it in the air, and we would measure the volume and things like that. And what happens is when your hand's down below your heart, gravity, right, is going to help pool your blood in your fingertip. And as the blood pools in your fingertip, we're going to see a bigger volume, which means we should see the pressure wave go up. When your hand is above your head, we would have done that next. we would have seen that with your hand above your head, there's a decrease in volume because the blood is going to drain out of your hand, and that's going to decrease the pulse pressure wave. In either case, though, the ECG size doesn't change because ECG is electricity, and electricity is unaffected by gravity, by body position, while the pulse volume will be altered by the position of your hand typically. So looking at this, when we measure the QRS amplitude, notice it's pretty much the same all the way across, right? So this is the electrical activity we're measuring. We're measuring the height of the, basically from the lowest point, often the S wave in this case, to the highest point, which would be the peak of the R. It really doesn't change any. But if we're measuring the relative amplitude of the pulse wave, if we're measuring from the peak of a pulse to a peak of a pulse, we'll see that it goes up when the arm is down because blood is pooling in the hand, and it goes down when the arm is up, and sometimes it completely goes away because blood is draining out. It depends on a couple other factors as well. So kind of looking at this figure, we can kind of look at a couple things, all right? So when I was talking about the QRS amplitude, what we would do is we would measure from the lowest point there and the highest point there. And that difference right here, that would be the amplitude of the QRS. And that, looking at the previous slide, didn't change much. What we're looking at here, this is the pulse pressure amplitude. So notice how big this is. And this is related to the volume of blood, the size of it. So when your arm is up, this is going to get really small. It's going to look something like this or even smaller because blood is draining out of your fingertip because of gravity. This is somebody with their arm down, to be honest, from that. And I can tell because how big this pulse pressure amplitude is. It's very, very large, relatively speaking. So one of the things that we also want to look at is we want to be able to calculate what the distance the pulse pressure wave travels, okay? So what we do is we measured how far from your heart, basically, to your fingertip did the pulse pressure wave go. So what we did is we measured from somebody from the heart, so basically from there, out to the shoulder, and then from the shoulder we straightened the arm and measured the length to the fingertip. So in the person, and this is not the person, this is from BioPack itself, the person we had was actually a fairly small person. Their distance between their heart and their fingertip was 76.2 centimeters. Okay? So that's how far it was from the heart to the fingertip. Okay? So we know that. And then we also measured in different times, under different circumstances, how long it took for that pulse pressure wave to get to the fingertip. Okay, and what we did is we measured the peak of R, and you can see this highlighted area here, right? So this highlighted area right here, we measured from the peak of the R to the peak of the pulse pressure wave. Okay, and that from peak to peak is the transit time. It's how long it takes for the pressure wave to get from the heart. So that's when the heart starts contracting, right? So that's the beginning of systole, to actually being able to see the peak of the pressure wave. And so in the first instance, when the arm was relaxed, it was 0.34 seconds. Okay, and so knowing that, right, we can figure out how what the velocity of that pulse pressure wave is. So if we take 76.2, that's the number we get right here, right, centimeters, divided by 0.34 seconds, that gives me a speed of about 200, and it's impossible to write like this, but that's not, you can see that, about 224 centimeters per second. So the pulse pressure wave is traveling that fast. Well, when we take the arm up, like this person is right here, right, so their arm's up, the actual pulse pressure wave, right, has to travel from the heart, but it has to go uphill. It has to work against gravity. And just like if we timed you running a race flat and then timed you running a race uphill, you're going to go slower because of gravity. So in this case, we'll use 76.2, all right? Again, that's how far it has to travel. It's not going to change whether your arm's up or down or to the side. And this time it took 0.37 seconds to get there. It took longer to get to the fingertip because it's going against gravity. So in this case, it was about 206 centimeters per second. So the speed of the pulse pressure wave decreased because it's going against gravity. And so that's expected result. And it's just, kind of interesting little thing that we can do there. So that was that. And I believe this is the last slide, if I remember right. So I think we're done. And that should be the discussion for Lab 24.